RT-PCR RNA extraction. Overview. RT-PCR RNA extraction will be discussed under the following headings: Introduction, Components, Lysis Buffer, Silica membrane-based RNA extraction Part 1, Silica membrane-based RNA extraction Part 2, Magnetic bead-based RNA extraction Part 1, and Magnetic bead-based RNA extraction Part 2. Introduction. RT-PCR is the most widely used test to detect COVID-19 infection. This involves 4 steps: sample collection, RNA extraction, RNA amplification, and RNA quantification. In the previous video, we discussed about the nasopharyngeal sample collection. In this video, we will be reviewing the process of RNA extraction from the collected specimen. Components. The RNA extraction from the specimen consists of 3 major components: cell lysis to disrupt cells or tissues. This step helps in releasing RNA in free state. It is done using a lysis buffer. RNA concentration. This usually requires use of various solid phase extraction techniques. Finally, RNA isolation. This is done using elution buffer. Lysis buffer. For lysis, the tissue sample must undergo a treatment to break the cell membrane and free the nucleic acid. Nucleic acids are much more resistant to denaturation than most proteins. Therefore, while lysing cells for extraction of RNA, a denaturing lysis buffer can be used. It is usually composed of phenol and guanidine isothiocyanate. Another challenge is to protect the released RNA molecules from RNase enzymes. Therefore, RNase inhibitors are usually present in the lysis buffer. There are several ways by which RNA can be extracted from the specimen, and there are many different RNA extraction kits available in the market. Here we will briefly discuss about 2 solid phase extraction techniques: Silica membrane-based RNA extraction and magnetic bead-based RNA extraction. Silica membrane-based RNA extraction Part 1. This method relies on the fact that RNA will bind to the solid phase of silica membrane under specified conditions. This is the spin column containing silica membrane in the middle. When the lysate is loaded into the spin column, the silica membrane tends to selectively bind the RNA, and the remaining solution is collected in the tube. Next, when elution buffer is loaded into the spin column, it allows RNA to leave silica membrane and return to solution. Silica membrane-based RNA extraction Part 2. Here, the individual steps, which may vary between different kits, are discussed in greater detail. Lysis is achieved by adding lysis buffer. A buffer solution is added to the lyse sample, which provides right ionic conditions and facilitates RNA binding to the silica membrane. The resulting solution is then loaded into a spin column, and the column is put in a centrifuge. The centrifuge forces the solution through the silica membrane that is inside the spin column, where RNA will bind to the silica membrane. Rest of the solution passes through and can be removed. To wash and remove impurities, a wash buffer is added onto the column. The column is put in a centrifuge again, forcing the wash buffer through the membrane. This removes any impurities from the membrane, leaving only the RNA bound to the silica gel. Generally, it takes several washes until the RNA on the silica membrane is free of contaminants. Elution is the process of adding an elution buffer to the column. allowing the RNA to leave the silica membrane and return to solution. Then a centrifugation is carried out. This forces the elution buffer through the membrane. The elution buffer removes the viral RNA from the spin column and a purified RNA is obtained. Magnetic bead based RNA extraction Part 1. RNA extraction using this method relies on the magnetic beads which are specially functionalized to bind RNA. Magnetic beads are made up of tiny particles of iron oxides such as magnetite. Their unique surface coating and chemistry allows them to selectively bind to RNA. These beads exhibit magnetic behavior only in the presence of an external magnetic field. When the beads are mixed with the lysed sample, they bind RNA. When this solution is placed in a magnetic field, the beads with RNA will be held in place while the rest of the solution can be removed. Magnetic bead based RNA extraction Part 2. Here the individual steps, which may vary between different kits, are discussed in a greater detail. Lysis is achieved by adding lysis buffer. Then isopropanol is added to the lysed sample, which facilitates RNA binding to the magnetic beads. Magnetic beads are then added to the tube, which selectively binds to the RNA. Next, an external magnet is used to immobilize the magnetic beads with their attached RNA. The supernatant is removed and discarded without disturbing the beads. For further purification of RNA, external magnet is removed and a wash buffer is added onto the tube. Again, an external magnet is used to immobilize the magnetic beads with their attached RNA. The supernatant is removed and discarded without disturbing the beads. Elution is the process of adding an elution buffer to the tube after removing the external magnet. This allows the RNA to leave the magnetic beads and return to solution. Next, an external magnet is used to immobilize the magnetic beads, but this time without any attached RNA. The supernatant containing purified RNA is removed and it is collected in another tube. Thank you.